My name is Kim Bussey. I am an assistant professor in the same division as Buder, the Integrated Cancer Genomics Division up at TGen in Phoenix. And my background is I'm a cancer-sided geneticist by training, and went and did my postdoc with John Weinstein when he was at the NCI, and became an applied bioinformatician. And what I will tell you that means is I know how to use tools and I can figure them out really well. Do not ask me to program a thing. And today I'm going to talk to you about mining the gene expression data that is online and available in TCGA. We're not going to touch on downloading data sets, okay, because we've already talked about that. The pathway to do that is the same regardless of what kind of data you're after. And the downloading of data sets out of TCGA, they come in that tab delimited file per sample, which means you do have to work with them to get them. I will say that for gene expression data, particularly the array data, you can get raw cell files. And there are some tools that will allow you, if you want to download those raw cell files and work with that data, where you just create your download archive as a zip file, and then you go to the tool, and I'll explain it, and you open it up and say, pull this in and make a nice spreadsheet for me, and it does it. It's great. So the outline of my talk is I'm going to remind you a little bit about the biology. What do we mean when we say gene expression data? And give you some background about the types of gene expression measurements that can be made. We'll talk a little bit about what kinds of gene expression data are in TCGA, and then we'll focus mostly on the tools for working with that data. And the practicum will be actually going through, and you can follow along, and we'll go through working with some of the data, and I have some questions of pitfalls of, okay, now you've pulled it up, tell me what it means, kind of thing. So what do we mean by gene expression? So even though the central paradigm, central dogma of biology no longer holds one gene, one RNA, one protein, we still do DNA to RNA to protein. And when we're talking about gene expression, we are talking about measurements of RNA or protein. The platforms that you can have that are RNA-based can be categorized as array-based. These include Affymetrix, Illumina, and Agilent. They also include, if you've been around in the field for a while, cDNA spotted arrays. They can measure total mRNA. They can be focused on exons or splice variants. They can be focused on microRNAs. The probes are designed to specific sequences, and this is really important to keep in mind. You are not measuring everything. You are only measuring what's on the array. And that's important when you go to interpret the data. Then there is now RNA-Seq, which is sequence-based measurements of RNA. And it can be total RNA, it can be mRNA, it can be microRNA, it can be a targeted data set. It all depends on what you do to create your library. So an RNA expression workflow begins with RNA extraction, and you're going to have different protocols depending upon what you want your starting library to be. So if the target pool of RNA is total RNA, so you want to get everything, you're going to do it in a different extraction protocol than you might do for mRNA. So for an mRNA, you're probably going to do a poly-A enrichment, which means you're going to miss a lot of non-coding RNAs. So you need to keep that in mind as you think about the information that you are working with. And microRNAs require an entirely different protocol. You either create a cDNA or a cRNA library depending upon the technology. And for the array-based method, you have a hybridization and image analysis. And this has been pretty well covered, so I'm not going to go over it in depth. For RNA-Seq, what you're doing is you're doing a fragmentation of either poly-A or ribosomal RNA depleted RNA. Then you then create a cDNA library with the first and second strand synthesis. You repair your ends, you then add a overhang, and you ligate your adapters. And this is for the Illumina technologies. And once you have that, you have your PCR, you can clean up your bead, and now you're ready to sequence. So once you have your library prep, and this is DNA or RNA, you then do what's called cluster growth on your bead, where you're doing bridge PCR. And so now you've enriched for sequences on your bead. You're then going to actually go through the sequence. And this is what you get, is an image analysis. And what you're actually then reading out is the order of the images of the chemistry, the fluorescence. So it's actually an image-based system. For gene expression platforms, the protein, what's in TCGA right now, is reverse phase protein arrays. Okay, these are serial dilutions of protein lysate that are spotted on an array, and then the array is probed with an antibody. This is opposed to, you may have heard of antibody arrays, where you actually spot, you can spot different antibodies on, and then put a single lysate and see which binds. And so there are two different ways of getting at expression information. You may run into the literature things like proteomic methods that use mass spec, but those aren't in TCGA right now. So the process for reverse phase protein arrays begins with your extraction. And so you then denature your protein in SDS sample buffer. You do a serial dilution of your samples, and you array that serial dilution on a nitrocellulose-coated slide. You then probe the slide with an antibody. And it's important that the antibodies that you probe with actually only give you one molecular weight when you are probing, so that you know that you're being specific. So quite often for optimizing these, you have to do a lot of westerns to find the right antibody for the protein that actually will give you a signal that you are reasonably sure you're only measuring one species. You can get phosphospecific data, and in TCGA there is both total protein and phosphospecific, but the requirement for that specificity of the antibodies means that not every gene is going to have a measurement, and for sure protein expression is the least number of genes covered. So the TCGA data sets. This is the link to the data primer. This is your new best friend, because you won't remember this 2 days from now, and it is a great place to start going and looking through of, well, I've got this file and I can't remember what this header was. Keynote, remember that what you can access publicly depends upon the risk of being able to identify the subject. So particularly for RNA-Seq data, you will not be able to access BAM files. You are not going to be able to access FASTQ files without already having controlled access approval. However, if you're looking at an array-based, like I said, an array-based measurement like an Affymetrix chip, you can get the cell file, the .cell file. So you're familiar with the portal. The tools that are available here are a great way to interact with the data, and we'll go through what some of the tools can do and how you use them for expression data. But I want to back up here and take a moment, because the whole idea of this exercise is not only to be able to use the expression data, but to integrate it. So integration is the goal. And when I was a postdoc, I was working on the NCI-60 data set. So this is a slide from way back when. And the advantage of doing integration, the advantage of having many data points, is that you can have, for example, in the NCI-60 data, they have a drug screen. And then they have measurements, biological measurements. And then you also have databases of chemical characteristics of that drug screen. And this allows you to be able to, is my pointer, no, allows you to be able to trace a pattern in here of activity either across drugs if you wanted to go into molecular descriptors, or across cell lines if you want to go into biological descriptors, and look at things like protein, RNA, and DNA. And you can access this information if you have these similar measurements in whatever new sample, which includes TCGA. So the NCI60 cell lines were profiled several times on Affymetrix expression arrays. So if you're working in TCGA and you want to look at you've stumbled upon a signature in gene expression data and you say, hey, I'd really like to trace that into potentially an activity, a drug response profile, as a first step of translation, as long as it's the Affymetrix data, my pointer is it, then you can do that really easily. There are other ways to do it by generating a gene set, but it's much easier if you've got same platform, then this becomes really easy to do. Things you need to be remember about this. Integration is the goal, but you need to understand what the technology measured, okay? Array-based technology is a probe-based technology. And depending upon where that probe was designed, you may or may not have unique species identification. So Affymetrix, the U133+2 arrays, there's a subset of probes that are specific for specific transcripts, and then there are a subset of probes that catch every single transcript coming off a gene. They won't agree with each other. And you need to know that that's what that got measured so that when you go into RNA-Seq and you do your analysis, the probes that you're going, okay, I think maybe, I want to look at this. Why is the RNA-Seq data not jiving now that I'm looking at isoform or I'm looking at this? Well, you need to look at RNA-Seq data that actually looks at the total expression off the gene if it was an Affymetrix probe that measured every single transcript coming off the gene. You also need to know how that measurement was annotated. As part of my postdoc, I was lucky enough to be part of the development of a tool called MatchMiner, because back then we didn't have a completed draft of the genome. You couldn't annotate things by genomic coordinates well at all. And so you had a gene and you had to figure out, okay, I've got a DNA-based measurement here, I've got an RNA measurement here, this is annotated by backclone, this is annotated by Affymetrix ID. How do I tell what's on this chip that matches what's on this chip? Because they weren't arrayed in the same place, you can't do a coordinate matching. Keep in mind that you still need to know that. Even though our genomic coordinates, we're now annotating by genomic coordinates, you're going to need to know whether the coordinate in the data set that you're working on in TCGA was annotated against human genome build 18 or build 19. Because the coordinates differ, and they differ by about 4 megabases or more, and it depends on where you are and which chromosome. So you need to know how it's annotated. You also need to remember that not all identifiers are stable over time. For every bioinformatician in here who is going to hand back data to a biologist, I realize that the tools for sequence-based data are annotated with ensemble IDs. Okay, they're going to give you an ensemble transcript ID, they're going to give you an ensemble gene ID. Those are not stable IDs from build to build. The stable ID in that annotation is a RefSeq. You need to return a RefSeq ID to your biologists. And if it means that you need to go and do some additional annotation, please go do additional annotation, because that RefSeq ID ties back to a deposited sequence. And that's what got placed on the build of the genome. The ensemble ID did not get placed on the build of the genome. The ensemble ID was assigned once the ref-seq was placed. So keep that in mind. It also means that anytime you're doing this kind of integration and annotation, you are stepping into a moving river. Okay? It is not uncommon to do some work and then to have to write a grant that has nothing to do with the work that you're doing, and to come back 6 months later and go, I'm going to pick this up again, and discover that 10% or 15% or 50% of what you've done has changed. Okay? If that's going to drive you crazy, get a freeze of the information that you're mining from at the time you're mining from it. And then, as we discovered today, Excel does bad things to gene symbols and clone IDs. So as a reminder, and I'll go over this because it was asked, you know, kind of how do you do this on a PC, since we were shown how to do it on a Mac. It's pretty similar, but just as a reminder, Excel, you need the wizard to be able to import a text file to have control over how Excel is going to treat that data. If you don't do that, then Excel is going to try to be smart for you. And there was a question yesterday about copy number, and since I'm a cytogeneticist, I just wanted to expand a little bit on it. This is a normal cell. This is a normal karyotype. This is what you see under the microscope, okay? This is cancer. One thing that should be dramatically apparent to you is that there are way more chromosomes in the cancer than in the normal cell, okay? The other thing that should be apparent, that you may not, if you sat here and counted, this is not twice this. This is on, I think this one was roughly, I want to say just shy, hypotriploid, so just shy of 69. Okay, and then it's not normal. This part here of this chromosome is chromosome 6. This is a relatively normal chromosome 6. The normal chromosome 6 over there, where is it? Oh, come on. There is a normal chromosome 6 over here somewhere. It's just not jumping out at me. But you can see that here's 6p. Here's what looks like 6p, although this p is still not normal. It's probably 6 down to about here, and then I'm going to guess it either went to 12 or 1. I'm not sure because banding's not always great. It looks kind of like this, so it's probably chromosome 1. But this is a translocation. What this has done, here's one normal chromosome 1. I don't see another one right obviously. Is that this is how for this region of chromosome 1 you can all of a sudden go from saying, oh, the copy number is 2 to, oh, the copy number is 1.5. Because I can tell you that while this is a cell and a representative cell, this happens to be an adrenocortical carcinoma cell line. When I went to write up the karyotype for this, I had to write it as a composite karyotype. And what that means is that I had no single cell, and I counted 20. I had no single cell that was exactly the same. Okay? So that's how you end up with 3.7 for a copy number. The other thing to keep in mind, too, on copy number is that particularly for array CGH type measurements, and to some degree for the CNV, although they try to adjust for ploidy, the ploidy is assumed to be the relative average ploidy of the cell. And so what one copy up and one copy down means when something is 4 is different than what it means when something is diploid. And so you need to keep in mind that your CNVs are relative copy number changes. They're relative to the average ploidy of the tumor. And you won't know the average ploidy of the tumor because we didn't do the measurement. All right, so tools and questions that you can use. So you can use, these are all tools that you can link to. I personally download IGV onto my desktop, and the reason I do that is it's quicker to launch it from my desktop than it is to go to Broad and launch it, particularly if Broad servers are busy. Gene Pattern is the same way. Bring that one, a local copy to that. So IGV is a great way to visualize the output of Firehose. You can then go and go explore the data behind that through the portal that you were introduced to yesterday. It's also a great way to take advantage of, oh, I'd like to overlay the ENCODE data. I'd like to take a look at just general normal gene expression for my tissue of interest. Those data are available to you in IGV as a server query, and that's where you're going to find the TCGA data. You don't have to download a thing, you just have to get IGV running. The UCSC Cancer Genome Viewer is a great way to visualize and do subset analysis. So you can visualize data sets, and then it's got a very facile way to actually start to take a look at data based on a subset analysis. And you'll get a basic, well, as I'll explain and we'll touch on, you get a basic clinical subset, but it's a good way to, you can always add additional clinical features. So as the data is available, you can go in and say, okay, I'd like to subset, not just on by site of where the tumor was it primary or not, but I'd actually like to do a survival analysis. Regulome Explorer does integrative analysis, and it's driven by statistical associations. I think it can be very powerful. I will tell you that I have found it kind of clunky in that it is slow to load. That could also be the internet connection that I've had recently. And it takes a little while to figure out exactly how to use it, but once you figure out how to use it, it is good. And gives you information that nothing else, no other tool will. The Gene Pattern is a suite of tools that's offered by the Broad Institute that is available for many types of data sets. It'll handle methylation data, for example, if you want to do data analysis on methylation data. It is open source and it's free, so if you don't have access to something like Genome Studio, this is a tool that will do those analyses for you. There are canned pipelines. It has pre-processing tools available. One of my favorite is, it does take a particular type of file format. It's pretty easy to set up in Excel or anywhere where you can set up columns and rows. But for things like array-based data, particularly Affymetrix data, they have all the platforms already loaded. They have this thing that's a cell to .gct file. It's you just say, okay, here's my zip file of all my input data files. Here's how I'd like the data normalized. And here, and you can download an archive from Geo for the Gene Expression Omnibus. And you can say, and I only want 50 out of the 100 files in this archive, so you can specify, and it will build your input file for you. Which is great because it means you don't have to sit there and go cut and paste and cut and paste and cut and paste, and did I miss that one? And it's very nice. And then there are several data tools that allow you to do what's known pathway enrichment analysis. So IPA, Ingenuity Pathway Analysis, and GeneGo are the ones I'm most familiar with. They also require a license, so they're not freely available. But there are some that are, I'm just not as familiar with them. So out of the things that you can do, I wanted to highlight Biosig. So this is one of the tools from TCGA, and somebody yesterday said, can I actually see histology? Yay! Yes, you can. So this is how you find those images. Once you know what you're looking for, it's pretty easy to query. You can grab input sets, like most of the tools that'll work with TCGA data. You can either grab entire data sets or you can grab specific individuals, and you can see the histology slides. There's cBio, the genomics portal. This is a great way that we discovered is if you want to do across tumor type comparisons, this is a really good way to grab that data, because you can see you start out with all cancer studies. And so if you're interested in a particular gene or a particular subset of genes and how they're behaving across different tumor types, this is a good tool to be able to go and find that. Many of these tools are going to ask you for signatures or gene sets, and they are pre-programmed with several, but they may not be the ones you're interested in. This is a database that feeds GSEA, which is a tool that is also standalone, but it's found in Gene Pattern. It's gene set enrichment analysis. It's another way to do pathway or gene set enrichment. But you can mine the MSig database for gene sets. And they have different kinds of gene sets depending upon what your question is. So if you're actually interested in, say, I'd like to look at a specific region of the genome, you can mine positional gene sets. There are curated gene sets, which are the pathway gene sets. So they are KEGG. They are various databases. There are motif gene sets, which are based on the cis regulatory motifs that are conserved between human, mouse, rat, and dog. There are computational gene sets, which have been defined by mining large collections of cancer-oriented microarray data. These gene sets are the ones that sometimes make a lot of sense, and sometimes you go, There are the GO gene sets, which are the gene ontology gene sets. These can be really broad or not broad, depending upon where you are in the ontology. So just as a reminder, the gene ontology is an ontology to classify genes by biological function, by their location in the cell, and those are the ones I pay attention to. There's one other one. But up at the top is really broad. It has everything. The further you get down into the ontology, it is a diacyclic graph, which means that it is a hierarchy, but it is not a true hierarchy in that children, so down lower in the hierarchical tree, there's more than one way to the top. Children can have more than one parent, okay? And so my recommendation is if you're going after gene sets, to be as specific as possible when you're searching that, because it's really easy to come up with a gene set that sounds reasonable and has 500 things in it. And then there's oncogenic signatures, which are defined directly from microarray gene expression data from cancer gene perturbations. And so you can mine. The search will require that you, in order to do that, you have to register. Basically, you just have to give them their email address, your email address, so that they know how many times you come and take a look around in the database. It's a usage statistics thing. The UCSC Cancer Genomics Browser is a great thing. What it gives you is this data on one side and clinical annotations on the other, along with any subset analysis that you're doing. There's the Cancer Genome Workbench, which allows you to probe some data related to heat maps, as well as integrated tracks. So if you kind of want to do uses a specialized version of the UCSC general genome browser with specialized tracks, and you can query for genes and things based on that. And then again, IGV. And so this is the kind of data that you can pull in IGV. Like I said, it's a great way to look here. So what I did in this screenshot is I was looking at breast cancer data. In this column here indicates the same kind of measurement. The platform is indicated by the next column over. And so we have copy number, we have microarray data, we have methylation data, and then I layered that on top of some of the data coming out of the ENCODE project, which looked at the prevalence of histone marks by chip, which is chromatin immunoprecipitation, for histone 3 lysine 9 trimethylation, which is a mark of tends to be associated with inactivity. And so you would expect it to show up potentially with sites that are methylated. Was part of the reason to put it in. And then I added down here at the bottom, I added in the gene expression data from the Body Atlas project. So this is Illumina HiSeq RNA paired in gene expression data, so you can get splice variants. And we're looking at E-cadherin and a region just up of E-cadherin. And you can kind of see how in these two samples there's no copy number change. This sample has a loss. Then as we move into the gene expression data, this sample is actually relatively, well, actually, I'm not interactive, so I can't left click. When this is interactive, you can left click if you haven't turned off the mouse over function and pull up what the expression is. I believe the gene expression data is, no, it's coded the same way. So it's overexpressed. And then you have methylation data. Green is up, red is down. And you can see that we have a region of heavy methylation here. And so as you pull things in, you can see, you can match up identifiers. You can change the tags as you're exploring this, so labels. So one of the things that I discovered was that it was really hard to keep straight what I was looking at where last night as I was playing around more with this data, and I went in and said, okay, I got to label the methylation. I can't tell where it is. This isn't working. So I changed the label so that I just removed the TCGA part of the tag and said, yeah, I know it's TCGA. Here's what it is. So you have that flexibility as you can do this. You can export your views from here. You can reorganize panels. The one thing that I will say is that in as much as you can, run it on a machine that runs as most memory as possible, because the more integration, and have the largest screen you can possibly have. Because the more data you want to look at simultaneously, the slower it gets with the more data. And eventually it just kind of chokes on you and says, I'm out of memory, I'm not loading anything else. And it says it very nicely like that. But it's a good tool for that. So I'm going to leave us plenty of time to kind of go through the practicum, because the take-home message is play with the tools. There is you can't break them, okay? The other thing that I'm going to say is I want to give us a little time is because one of the tools, if you do not have Firefox, particularly if you're running a PC, one of the tools we're going to use, the UCSC Genome Browser, the Cancer Genome Browser, has known issues with Explorer. So Firefox is the preferred browser for that. So if you don't have it downloaded, please download it now. It should download and install pretty easily. But the take-home messages are that gene expression data can be RNA or protein-based measurements. You can integrate them. Different tools are good for showing you different relationships. And so get a feel, play with each tool and get a feel for what kind of relationships it shows you. And then the interpretation of the results requires an understanding of what was actually measured. And the practicum we're going to go over is actually going to highlight that for you. Okay? So if this, I'm going to warn you, I've played in here now, so my screen's going to look a little different than yours. If you haven't gone to this tool before, when you click on the Cancer Genomics Browser, if this is your first time here, you're going to get a little green window here, or yellow window, that's a tutorial, okay? Click on the little blue X. But you are welcome to come back and do the tutorial, and I would highly recommend doing it. It's a nice interactive tutorial. So it will say, to do this, you need to do this, and you can sit there with your little box open and then move around on the website. So I'm going to click out of the lung cancer. Okay. So a little bit about this. You start in a chromosome view. You can switch to a gene view. Most of the work that we're going to be doing will be in gene view. We're going to look and use the ovarian data set today as an example because it has just about everything we want to look at. Okay? And you can see here are all the data sets that are available for you to use. We're going to use, go ahead and load the AFI data. We're going to load the adjuvant data, the second adjuvant data, and the RNA-Seq. And highlight them and load them, and the protein data. Okay? And it'll take it a while. Yeah, AFI, adjuvant, the RNA-Seq, and the protein. So we're going to look at all of the gene expression data sets you could possibly pull. Okay, so you should be looking at something like this. You won't have this column yet. I have it because I've been in here, but you won't have this column. So let me explain a little bit about, these are the data. They're controlled here. You have 3 different options. You can look at a heat map, you can look at box plot, and you can look at proportions. You can change your coloring. So if you're not colorblind, you can do red-green. If you dislike looking at white, you can do yellow-teal or cyan. So you can change this for however you want to visualize the data, and depending upon what you're looking at, you may find that you move from, say, heat map into proportions, and you're having a hard time kind of seeing gradation, and it's easier to switch things over. This is your clinical annotation, and what normally comes up is just basic sample type, things like grade, age at diagnosis, days of follow-up, and the sample names. So if you want a subset based on sample name, you can do that. If you want to subset on days of follow-up and things. So this is, you've got your protein data, and we have our gene expression data sets. And you can notice that each platform has a different number of samples. Okay? Some platforms have more, some platforms have less. Okay, and you go all the way down, finally to the AFI at the bottom. Okay? All right. If you want to add the days to death, or to add, if you don't come up, for example, age of diagnosis doesn't pop up. If you click on the plus, these are all the additional clinical features you can add. So you can see what different things you can put in. All you have to do is highlight them, and you can see. see them. They must be highlighted to show up in your subset analysis. So where you get your subset analysis is in the little wrench. And anything that you apply for a given cancer set, so if you're looking at ovarian cancer, anything that's ovarian coming out of the ovarian cancer data, any subset you apply here will automatically apply to all of the other things you have open. Okay, so you can see it all at once. And until you turn it off, it's going to give you a little reminder. Hey, I'm applying this to everything. So if that bugs you, feel free to turn it off. All right, I'm trying to remember. Okay. So this, we'll come back to this in just a minute. We're at the chromosome view. You can look at the gene view. And what I have highlighted here, the way you get a gene view is to pick either a signature or a gene set. So right now we have the IGF-1R pathway highlighted. There are, this is where you would find it. If you want to remove it, you click the little blue X. Okay? And this has lots of interesting things. And so one of the questions that you might want to ask is, does age of diagnosis influence the activity of this pathway? This is an important pathway in cancer. It drives growth. It drives survival. And in fact, IGF-1R actually drives a pretty even distribution between growth and survival, geared a little bit more towards anti-apoptosis, as opposed to the MEK-ERK, the MAP kinase pathway, which drives heavily proliferation and not quite as much survival. And why that is important is that this may play a role in how sensitive things are to therapy. So coming into this, we're actually going to look at age at diagnosis, and we want to look at patients that are less than 50. So you slide to the best of your ability there. The advantage of this is that it computes these funky numbers are actually represent the actual data points. So when you see 495, yeah, the next one up is probably there. And you can add this. Now, it's going through and it's doing the subsetting, but it's also brought up this really cool thing. I know enough statistics to be dangerous. I do not know enough to explain all of the tests on here. Please read the help if you want to use more than like a student's t-test. Difference in means student's t-test, Wilcoxon, Fisher's exact. There are things that you can do to test for differences between your subsets. So we'll stick with the student's t-test, and because there are several, we're making lots and lots and lots of comparisons, we're going to do an FDR, Benjamin Hockenberg correction. Okay, Bonferroni is a much more conservative correction, as I'm sure most of you are aware, but it tends to, particularly when you're starting to explore data, throw the baby out with the bathwater, and you miss a lot of really interesting things. So at this point when you're doing data exploration, you're generally willing to take things that a false positive rate of, say, 20%. I'm willing for 20% of my hits to be wrong, because 80% of the time it's going to be worth following up. Okay? And then click Show the Statistics. And it's gone through here, and it's given you statistics here. So it's actually telling you where there are differences between the subsets, And the fact that these are gray mean that they don't actually qualify as being statistically significant in one subset or the other. And now that I'm looking at this, I'm going, this may not be the right pathway I'd like to look at. So I'm going to come over here, and we're going to get rid of that pathway, and I'm going to grab the PAM50. It has more genes. It automatically updated my statistics. And all of a sudden, now we have comparisons. Okay? Has everybody been able to get on now and interact? Or are we still waiting to get to the website? Yes. Click on gene. So up here, make sure that it says genes.
Oh, I see.
Yeah, it took me. I was like, why can't I get the O? Okay, and then it'll be under the add favorites. All right. As you look down, you'll see that we assigned it for protein, but we didn't assign it yet for the expression data. All right, so we don't have the comparisons done for the expression data, the mRNA level stuff. So go into that and tell it, you don't have to reassign these. The assignment's already been made. You just need to tell it to show you the statistics. So go through and do that for every data set. Come on. That one has it. Am I done? I am. Okay. So what do you see as you've got those set? Take a look at the data. It's the same set of genes in different measurements. And what are you seeing with the statistics? So if you look, for example, at CXXC5, and you scroll down with the data sets, are you getting the same result? Why?
I actually just got my thing to display.
I mean, we can take an easy one. Why might you not see it in, is it even measured? I mean, it's not even measured in the protein, so that explains why you don't see it in the protein. But why might you not see it in, say, the RNA-Seq data, but we see it being grouped with this subgroup? No, that's not what I wanted. So the green, these tell you where it's enriched, where you're actually seeing a significant difference.
Which statistical analysis were you using?
I was using the student t-test with the FDR correction for the p-value. It's also fun to play with it with no correction. I mean, that'll show you some of the same things, but
Which set again?
ovarian cancer? The gene set, I'm using the PAM50, sorry. Yeah? Yeah? Yeah.
So I have everything displayed. I picked the gene set that you're talking about, the IGF-1 pathway mapping.
Yeah, so get rid of IGF-1, click.
Okay, I can get rid of that.
Yeah.
And then what's the question?
So the question after you've got the statistics up and running is, are you seeing the same thing out of all the data sets? And if not, why not? So the statistics again are, you go into this little mouse, once you mouse up here, you'll get the little wrench. You'll need to subset your data first as less than 50 age of onset or age of diagnosis, and greater than 50 at age of diagnosis. And you're going to compare with a student's t-test with the FDR correction on the p-value. So one of the things that you should notice as you're coming through here is that reasons that you may not be getting consistent results between data sets are, again, what did you measure? So I know for a fact that the Affymetrix probes for some of these genes measure all the isoforms the RNA-Seq may not have. But likely what's influencing it is the number of samples that are actually included and have the measurement. Okay, but the advantage of looking at multiple measurements is that if you look at every single one of these, pretty much MYC is up. Sorry, stop that. Progesterone receptor is up. ERBB2 is slanted the other way. It's down, so it's more prevalent here. And that is somewhat consistent across all the data sets. So even though you've done an FDR correction and you've said, I'm willing to get 20% of my hits are false positives, they're wild goose chases, if the signal shows up in multiple platforms, it's probably real, right? The other thing I want to show you here then is let's take a look at zooming in on the protein data for estrogen receptor, okay? Because I want you to see some cool stuff. There's more than one measurement here. So there's total protein and phosphoprotein. The protein data, so down the list, back here, down here.
How do you remove the feature like with the plus sign? How do you remove that data set?
How do you remove it?
Yeah.
You come over here to the feature, you click on it, and you say hide.
How do you remove the entire thing?
Oh, like the window?
Just like instead of Seq on the cross. Oh, okay, I know what you're talking about.
Yeah. So the protein, depending upon the protein that you measure, you may have total, you may have total, you have phosphospecific if it's a phosphoprotein, if it's something that can be phosphorylated. And so ESR1 can be phosphorylated, and so here's a particular reading for R5 serine 118. Here is R5 alpha, and here is M5 alpha, MV alpha. These are two versions of estrogen receptor, okay, that they've measured differently. And you can see that we have a significant difference between the two groups in ESR1 for total receptor expression, but we aren't affecting the phosphorylation status. Okay, so you can drill to that level of detail here. If you want to zoom back out, you come, no, wrong one. Yeah, scroll up. Something happened to my screen here. There we go. I can't find my scroll bar. You can, this will zoom it back out. Okay? Okay? Now we've done the statistics here, and let's, for kicks, let's say we'd actually like to add DNA methylation and copy number, because we'd like to see whether the gene expression for MYC is often amplified. Is MYC being amplified, and is estrogen receptor and lack of expression of estrogen receptor being indicated by methylation? So you can just add the methylation data. It's going to add it at the top. Okay. Ugh. Error has occurred. Really? Okay. Kill that. We'll try it again. And load the logistic too. And okay. You'll have to go back in and set the statistics, potentially. So I added the logistic 2 copy number, and I added DNA methylation. Okay. And you can see that our subsetting, depending on the set, actually kind of changes. because again, the number of samples with different measurements differ depending upon the platform. Now, if some of you are actually getting your statistics with the data sets, because it's not letting me grab statistics, such as the nature of live things, the question I have is, are the same things popping? Are the same genes popping? Or are you seeing some different genes come up in the data sets? And methylation versus what you saw come up as being, so remember we saw estrogen receptor, MYC, progesterone receptor, ERBB2 being things that showed some consistent patterns. Are they also showing consistent patterns in the 2 new data sets that you added? If you have a signature, it can evaluate a signature, and the difference in the tool between a signature and a gene set is a signature says, I need to see the expression levels of these 3 genes added together, divided by the expression level of these 5 genes, multiplied by something. You know, it's an algebraic expression describing what you want to see. And UCSC, this will evaluate that expression and then show you the results. And then you can switch. I talked about it, but we didn't do it. You can see the proportions. So this is actually kind of a fun, easy way to see what's happening in the cell line data or in any of the data sets. It gives you an idea of what the proportions are and their expression levels relative to the groups. Okay? So focusing on the genes that came up as we were looking at the clinical samples, one of the things that you can do is we define these relationships by age of onset. And it'd be kind of interesting to know whether there was actually a survival advantage to it. That might be the next question that you want to ask. And so the place to do that is here. And you're going to go and you're going to grab the provisional at the cBio. And you want to grab the provisional and I'm going to grab all the data and I'm going to accept the defaults because I have not played with the data enough for this particular data set to know whether those defaults are valid or not. This is why I say please play with the tools. I picked the ovarian cancer, the provisional data set, not the manuscript data set. And in here, I'm going to enter my gene names. I think it's 1. That's not it. PR. Progesterone receptor. And you notice that when I don't get the Hugo symbol right, it lets me know. Okay. So these were the genes that were having changes that were so much the same over multiple sets. And the complete tumors are the ones that have all the data. So they'll be looking at just those samples that have data for all the data sets that we've asked for. and you can see that we've got now Oncoprints. Okay. This is a good way to start exploring pathways. You know, you saw these were the PAM50 signatures. So you can look at the network. This is actually going to give you something along the lines of what you might get in IPA, and it's free. So it's showing you a network of genes. The genes that you put in are highlighted in black, and then it's showing you the connections between the networks. And you can go and explore what these are and the relationships between them. We have plots. And so you can do mRNA versus methylation for MYC, looking at MYC, protein level versus mRNA. That's not a half bad correlation. But you can then take, this one is going to give you a funky correlation. If you were to run a Pearson's correlation coefficient, this would actually give you a correlation because it's going to be driven by these outliers up here, but that really isn't correlated. Okay, this is protein level to mRNA level. This one actually is pretty well correlated. And this is progesterone receptor. Okay, so you can play with these kinds of things. There's our survival curve. So while there's interesting things, it's not panning out in survival. Mutual exclusivity. So again, looking at things that have trends to, you have almost a co-occurrence between MYC and GPR 160, which is an interesting finding. So things that kind of go hand in hand. You can look at mutations in the data at fine detail here. Yes. Well, okay, so keep in mind that here these are two different tools. They're designed to give you easy access to different kinds of analyses. So when you're switching between tools, the tools don't necessarily talk to each other. The advantage of the fact is that they use the same data sets, but the tools don't talk to each other. So there are other things that you can kind of play with here and around, in and around here. The one question that I have for you and I want you to do is to think about, we didn't see a difference in survival. So we started with looking at age of onset, and we picked some genes that were differentially represented based on age of onset, and we went to this tool which easily allows you to look at survival, and we don't see a difference. And it's quite possible because late onset, these genes don't play a role. Early onset, they do. And you would need to tease that out. That's a very valid point. But you've looked at it and you've said, okay, the important thing driving those genes is age of onset. So, given that, if you go back to the UCSC Cancer Genome Browser, can you figure out what the genes driving survival are? So that's your task. So when you go back to the Cancer Genome Browser, okay? And so we went through and we looked at age of onset. Can you, based on what I showed you, see if you can identify a set of genes that drive survival? Now I'm going to tell you, this is going to involve having to add a clinical annotation, Okay. Subsetting on that annotation. And then asking the question of what are the statistics. So that's what we did for the age of diagnosis. Okay. And because my browser has had an issue here. Let's see. If I load you, will you come up as now allowing me to do things? Okay, so looking at down this list, you're going to be looking for something that gives you an, keep scrolling. There's something in here that tells you about survival. Yes, days to death. And we can actually, in this tool, if you subset on both, early survival and days to death, we can then start to see days to death for me is already clicked. Yeah, I already selected it. I'm like, why is it grayed out? So
What's the maximum you have the
yeah. But you could, yeah. What is 5 years in days? 1800, roughly 1800 days. Yeah, something like that. Yeah. So you could say, okay, and say 2 years. So roughly. Oh, add.
My computer is now just watching.
And we'll see. I'm guessing here. This is slightly less than 2 years. And this is more than 5. So the young versus old covers most of the yeah. So the question here is as you're playing with this and discovering, you got to worry about over parameterizing, of dropping too many subjects, not having enough to actually make some sort of valid inference. And in terms of age of onset, when we did that, if you looked at the data sets that had almost 600, the vast majority of the tumors landed one place or another. They were classified one way or another, and it was a pretty even breakout. So I wasn't too worried about early onset. I'm a little concerned here about the breakout on the subgroups here, which tells me that I'm not going to be able to make a whole lot because days of the way I've grouped these, because I've grouped for extremes on survival, I've now got the vast majority of my data is in that intermediate range. So they're somewhere between not quite 2 years and 5 years in survival, which means I didn't divide my age right. And I'm not an ovarian cancer expert, so this is the advantage of this tool. You can look at that and go, oops, didn't do that right, and go back in and play with your subset.
Is it really that you're not an ovarian cancer expert, so it's just the sample set that exists in the database?
So the sample set that exists in the database, you know, yes and yes. The answer to that is yes, it's a lack of expert knowledge and the limitations of the sample set that you're working with. If you have expert knowledge, you will make better guesses the first time around. If you want to see, so this was the way I didn't remove, no, that isn't it, that's it. I didn't remove this young age, but if you wanted to actually say, okay, this is one way to say what that was driving age of onset, does it carry over into survival? But the other way to ask the question is, I just really would rather look at just straight up survival. And so you can remove age of onset. And again, this gives you a good representation of, I don't have a lot of long-term survivors. So again, it gives you a good feel for what your parameters are in your data. So for the rest of the time, play. Play with the tools. And I will try to, and Badur will try to help you. If there's a tool that we didn't cover that you're interested in playing with, feel free, bring it up. And if I don't know how to use it, I'll figure it out real quick. Okay? Yeah. That's okay. And so you're in GBM.
Here was where I selected what the 27 number. So maybe
To analyze.
Okay, and
That's a, that's
would I
a,
have to start processing? Do I have to
Yeah, I would, it doesn't like say me, you know, if it like you waiting for a relapse, like apply, right?
So I promised that I would show how to do this on a PC. So if you're running a PC and opening a text file with gene names wasn't something that you followed well from the Mac, it's under File, go to Open, navigate to where you're going to be, tell it All Files, click Open. This is a particular, so what it's done is it's put each gene in a column here. It's exported the gene sets that I selected from, and it's put them in rows. Let's see, those, come on. And then you get to go do this. I'm just going to quickly scan here and see if anything is going to get smacked. No March. So I'm scanning through here because if nothing's going to get screwed up by Excel, I'm not going to take the time to go click all of them. And then if this is the gene list you want, let's see. Come back over here. And if you want to be really savvy about this, whoops, that wasn't what I want. Okay. You can actually take it into a text editor, tell it to replace all the tabs with commas, and then copy that and place it in. But this should, well, let's see if it actually.
Otherwise, you have to just do it
Manually, yeah. So.
All these people that write scripts are like, I don't know why they're.
Why in the world do you do this? Because I don't program. And this may not, yeah, Notepad's, so Notepad isn't going to do that one. You'll need to,
The idea.
yeah. Word will? No, well.
Just trying to get the list of gene names with proper space.
Yeah.
It's already a text file,
Yeah,
isn't it?
she has a tab that file there. She wants to get rid of all of the spaces. She doesn't have a comma in between.
Replace this all the way on the right.
You could have, there's a couple ways you could have.
No. Okay, more. Special character. Replace. Tabs. How about all? Okay. There is a way to do it. Obvious. Mm-hmm. That's right. Tab character. Tab character, replace with comma, replace all. There we go. So now you have it. Oh. It's all right. We don't have.
We can find something out about colon. Very nice.
And so now you can look through. But if you wanted to take that, since you now have it,
Can I ask you an Excel question?
mm-hmm.
I always wonder, this gotta be an easy answer. If you want to switch, which ones are the rows and which ones are the columns?
Transpose. So paste special transpose. Now I'm looking at the provisional data, so that's why I have more access here. So the UCSC Cancer Genome Browser right now only has the published data. And we'll wait for it to pull all the data. Okay. It says, hello, don't like you. So, but here gives you an idea. Now, remember that the way I found this signature was to search for a pathway a canonical pathway that was annotated as colon. So we're pulling genes that are already known. So here are things that are mutually exclusive and inclusive. So you can go through and look and say, okay, you know, we've got several things that don't occur together, and again, some user notes to pass on as I'm playing with these tools more. I'd really like to be able to keep, when I'm scrolling across, this needs to stay here. So we'll pass that on. But you can see, for example, that KRAS is mutually exclusive with mutation in PI3 or alteration in PI3 kinase. So this is a case where it may be more beneficial to explore the data here first. Okay.